first step of tissue culture is excising or cutting a small part of plant tissue, like the leaf, root, or shoot of a plant. This cut-out part, explant, is then placed in a suitable nutrient medium containing a carbon source such as sucrose, inorganic salts, vitamins, amino acids, and growth regulators like auxins, cytokinins, etc. The explants are allowed to multiply and form a group of tissues called callus. Callus is irregular, unorganized, and undifferentiated mass of actively dividing cells. This callus is then allowed to multiply to form many plantlets, which are then transferred to a different container for rapid multiplication. Finally, the developed plantlets are transplanted to the soil for normal growth.
The world's first mammal to be cloned from cells taken from an adult Dorset ewe. To help you follow the process, we have labelled the parent sheep, sheep 1 and sheep 2. Note that sheep 1, the ewe to be cloned, has a white face, a fact that was used to help check that cloning was successful. The first step involved taking cells from the udder of sheep 1 and culturing them in a gel to create a batch of identical cells. In the course of culturing these cells, they were deliberately deprogrammed to let these cells regain their potential to become totipotent, to form any cell needed in the future clone, and not simply another cell from a ewe's udder. This single achievement marked a significant breakthrough. Meanwhile, an egg or ovum was removed from sheep 2, a black-faced female Scottish ewe, to be prepared for implantation. Using techniques developed in human fertilization treatments conducted at a microscopic level, the nucleus from the sheep 2's ovum was removed, or enucleated, to make sure that none of sheep 2's DNA and genotype could be passed on to the next generation. Once the ovum had been enucleated, it was then fused with a cell from the sheep 1 culture of deprogrammed udder cells. In effect, emulating the process of a sperm entering the egg, but with both copies of the chromosomes coming from the same parent sheep, sheep 1. So the newly fertilized egg is genetically identical with this single parent. Once fusion had taken place, chemical factors in the egg are released to stimulate cell division. Once the cells had multiplied sufficiently to form the makings of an embryo, the new cell cluster was transplanted into the womb of a third sheep, which we will call sheep 3, for a 21-week gestation period. In total, 277 attempts were made to fuse the adult cells with enucleated eggs. Only one survived. Dolly was born on the 5th of July 1996 by caesarean section.